So today we're going to talk about the plasma membrane. But when we talk about the plasma membrane, we're really talking about all membranes. So we have that endomembrane system, all that Golgi, endoplasmic reticulum, vesicles, lysosomes, peroxisomes, the nuclear envelope. When we talk about membranes today, we're not just talking about the plasma membrane, the one that surrounds the cell. We're talking about the composition of all membranes inside of a cell. And the structure of those membranes is often described as a fluid mosaic model. Now, a mosaic is something like a stained glass window, where it's not an individual piece of glass that makes the picture, it's all of the pieces of glasses together. Or if you've ever seen a daisy. A daisy, this looks like it is one flower. Actually, it's a composite of flowers. What you think of as a petal is really one whole flower. Every little circle you see inside that circular part of the daisy is also an individual flower. So when you look at a daisy, you think you're looking at one flower, but you're actually looking at tens to hundreds of flowers. That's what a mosaic is. And that's what we're going to look at when we talk about, or that's sort of the idea behind the structure of our membranes. And so the key here is we're going to be looking at figure 4.4. And if you look at figure 4.4, what you see is a collection of molecules. And it's not one molecule that makes the plasma membrane, it is the collection. And that's where the word mosaic comes from. So the predominant structure you see there is the phospholipid bilayer. So bi means two. And so when we look at this, we see a whole bunch of these lollipop-like structures. And we've already talked about phospholipids. And so we know what all these mean. So here is our phospholipid bilayer. Here's one layer. Each layer is called a leaflet. So this is going to be composed of two leaflets. And what's very important about this is the chemistry. Because the molecule, the phospholipid, has a certain chemistry, remember this upper part is polar, and these fatty acids are nonpolar. And over here, we have nonpolar, and we have the polar heads. A phospholipid bilayer will always orient in this direction. And that's because of the chemistry that's going on here. Remember, the term for something that's polar and nonpolar is amphipathic. That's why I introduced that word early in the semester. It's a word that I'll use a lot. Inside your cell, so remember your plasma membrane separates the inside from the outside. Inside your cell is cytoplasm. And most of what makes it the cytoplasm is water. And if we recall, water is also polar. So if we have a polar head and we have water that's polar, we're going to begin to build hydrogen bonds between these molecules. This is why the polar heads always orient outside. Polar forms hydrogen bonds with other polar substances. Same thing outside your body. Between cells, you also have water. So there's water molecules outside here. They will also form hydrogen bonds with those polar molecules. Polar things like or dissolve in other polar things because they're hydrophilic, i.e. they form hydrogen bonds. Nonpolar substances don't like polar things, so that's why the nonpolar polar substances orient towards each other. Nonpolar is going to bond with other nonpolar. And this chemistry is going to be important when we begin to talk about what kinds of things will be able to pass inside and out of a molecule. So, phospholipid bilayer is made up of two layers of phospholipids, and they will always orient with the polar outside, nonpolar inside. So this is what is going to separate the inside from the outside of our substance. Another thing that is going to be important in plasma membranes concerning this phospholipid bilayer, so that is the chemistry for why they orient the way they do. But also we talked about saturated fats and unsaturated fats. This will also play a role. In that phospholipid bilayer, how flexible it is going to be determined by, so flexibility is determined by fatty acid composition. So, a saturated fatty acid is a linear structure. So, when I draw a straight line, this is going to be a saturated fatty acid. That means it only has single bonds between the carbons and the hydrogens. If I draw a bent line, that would be an unsaturated. Sorry, I got a little cramp there. Because they have a double bond, at least one, then it will cause it to bend. And if it bends, then actually that gives us a little bit more room. If you are saturated, you're going to be really cramped because you can fit together much easier. But as soon as you throw in an unsaturated, notice all of a sudden we've got a little more room. So these saturated fatty acids are going to give us a lot of flexibility because the more room we have, the more wiggle room we have. This also can mean molecules can pass between some of these things easier than others. So, the type of fatty acid tail is going to give us more flexibility or less flexibility. Saturated fatty acids have less flexibility. Unsaturated fatty acids will have more flexibility. All right, so we've talked about the orientation of the phospholipid bilayer, how the phospholipids will orient with the polar heads out, the inner tails next to each other, and about flexibility depending on what type of fatty acid tail is involved. You'll see in the picture, the next most prominent thing are all those purple things that are running through, and those are proteins. So embedded in our phospholipid bilayer are going to be lots and lots of proteins. So let's draw that. Here's our phospholipid bilayer. I'll just put in a few phospholipids. And then you'll notice all of a sudden there'll be a protein. And it will span the entire length. If you have a protein that spans the entire length, this is called an integral protein. Other proteins will just be on one side, and those are called peripheral proteins. So this isn't seen in your book, but I'm going to have a diagram in your assignment today, and you'll be able to also see peripheral proteins in there. So proteins are going to serve several different functions, and these are seen in figure 4.5 in the reading next to figure 4.5. So what do those proteins do? So those proteins are going to help us to move things in and out. So one type of protein is called a channel protein. And this will help move substances in and out of the cell. However, these channel proteins, they're just big pipes. And so substances will come in and out, and they will have no interaction with the protein. So this is just like a pipe in your house. The water is going to flow in and out of your house through pipes. It's not going to interact with the pipe. It's just going to move through the pipe. However, there are some proteins where you will actually interact. You need sort of special passage. These are called transport proteins. So they have the same function. They're going to move substances in and out of the cell, but they will now interact. These are not just straight pipes. These will be composed in such a way that they will have a gate. And so the substance is going to have to come in, pay a fee. So that's a gate. So substances, as they come in and out, they have to interact. So they'll interact with the protein. And they will, like I said, in a sense, this is like a toll booth. And so they need energy to move in and out. Just like you pay money to go through a toll gate, these will use energy to open this gate, pass, or open the gate and pass this way. But they will need energy in order to pass. A third type and I'm just going through figure 4.5. So another type is called cell recognition. Oops. So number 3, cell recognition. This is an ID tag. It's like you having a driver's license in order to show who you are. This is important for a couple reasons. You need to be able to identify what are called your cells, your cell cells, versus antigens. These are foreign particles or cells. Your immune system has to be able to know what's you and what's not you. So cell recognition proteins, they will often have a carbohydrate attached to them. So these are often what are called glycoproteins. So they're protein and a carbohydrate. Anytime you see glyco, that means carbohydrate like glycogen and glucose. So this will help your immune system to say, hey, we don't want to attack these cells because that's who we are. But this is foreign. We need to get it out of our cells. And if you don't have a self-recognition protein, you'll be booted out. This is also helpful for organ identification. So we talked about hormones are chemicals that will be able to allow us to communicate. Well, if your brain sends a hormone to your liver to do something, how does it find the liver? Well, we have a cell recognition. So the hormone is going to go looking around for a liver cell recognition protein. And then it knows where to stop in order to send its message. So you can also think of this ID tag as like your address. You know, if I need to send something to my house, I put the address down and the postman knows exactly where to drop off that letter. All right, next. we have receptor proteins. So once the ID tag identifies things, where does that hormone then need to go? So you're going to have some of these. So I'm going to draw a little bit of a plasma membrane here. And often, sometimes ID proteins might also be receptor proteins. So here's my protein. And I'm going to so this one here might be a recognition protein, and then this one here would be a receptor protein. Notice it's got sort of a docking station. So that divot is a docking station. So if my hormone is coming along, and here we're going to put our little, this is going to be our glycoprotein. So here's the ID tag here. So as a hormone is traveling outside of this cell, so this is my hormone molecule. So as it's traveling along, it says, oh, I found the right cell. So now I need to dock onto the cell, so it finds a receptor protein, and it will fall into that dock, and it will then allow the cell to receive the message. So receptor proteins are, in a sense, docking stations for substances to interact with cells. Because then once this hormone docks in the station, this protein will change its conformation. It'll change shape. However it changes its shape, that's the message that comes into my cell. All right, 2 more to go. The next one that's in the book is an enzymatic protein. So enzymes are substances that help carry out metabolic reactions. So some of my proteins, they work best near a membrane, enzymatic proteins. So these are enzymes that work best in the area of the membranes. So they're just going to do their job, and these will allow us to carry out metabolic functions. So we'll talk more about enzymes in chapter 5 or 6, I can't remember which, but enzymes are just proteins that carry out metabolic functions. All enzymes need an optimal pH, an optimal salt content, an optimal water content, and so some enzymes, their best place to work is in the membrane. And then lastly, we have junction proteins. These are going to allow cells to adhere to each other. So that's pretty important. In order to make tissues the next level up, cells have to combine together. Junction proteins are one of those things that are going to help knit cells together so that we can make tissues. But also, it allows for cell-to-cell communication. Your cells will talk to each other to let each other know what's going on. One of the very important times that they do this, and you'll talk about this in micro, is if one cell gets attacked. So here's a cell, and if it gets attacked by a virus, it'll send a signal, it'll send a chemical to the next cell, and it can do that through these junction proteins to say, hey, I'm being attacked, you know, start defending yourself. And that way, if the virus tries to go to the next one, well, this one might be a little bit more prepared and have a better chance at breaking down that virus. But also, cells need to coordinate metabolism. So you might have a series of liver cells. And so they may need to talk to each other. We mentioned that glycogen is how you store carbohydrates. Well, this is often done in the liver. And so once this one's full, it's going to say, hey, you need to ramp up your production. And then once it's full, it'll tell the next one, hey, I'm full. You need to start ramping up your production. So cells need to communicate with each other, and they will often do this through these junction proteins. And then the third thing that you need to know for the different things in your membranes is you can see the molecule cholesterol. And cholesterol is a very important molecule. While we have to be aware of how much we get in our diet, it is still very important. Your body, however, can usually make all the cholesterol you need, which is why it can cause problems if you add too much into your diet. But the cholesterol is there. It's going to help give us structure to that plasma membrane. And also the cholesterol helps to add to that fluidity. So the cholesterol is going to allow these membranes to be fluid, and we know they're fluid because all you have are membranes. And so you notice the collection of my cells here, you can see how fluid it is because that's all I have around. Cholesterol is going to be one of those things. If you don't have the cholesterol in there, you can begin to get brittle plasma membranes and you can get cell ruptures. And it also helps to give you some stabilization with temperature. The cholesterol can help your cells stay together if it gets too hot or too cold. So that's what we mean by structure.